Welcome back, class. This is Living Systems. We are now in week 5, and tonight we're going to cover 2 very important lectures. The one that you can see here, chemical reactions, and then we'll move on to lecture 6, biological molecules. All right, so they're a little bit tough, tonight's lectures, a little bit tough. But rest assured that your tutorial questions will help you get through. And also, of course, we can always spend time on some of those questions over the next week, if you like. All right, so let's move on with the lecture on chemical reactions. All right, so you remember a few weeks back we talked about physical change and chemical change. In fact, that goes right back to week 1. Well, tonight we're looking at chemical change. More specifically, we're looking at chemical reactions. That is how one substance is converted into another substance. So chemical reactions involve breaking bonds in the starting materials, which we call reactants, well, in chemistry anyway, and forming new bonds in the final products. So let's look at a good example here of a chemical reaction, and this one is between CH4, which is methane, and oxygen O2. They are represented here. So here's one molecule of CH4 in black and white, and one molecule of oxygen O2 in red. Now, when those molecules react with each other, which they will in the presence of an ignition source, such as a flame, then a chemical reaction takes place where these atoms, the bonds between these atoms will break, the bonds between these 2 oxygen atoms will break, they will reshuffle, and new bonds will be formed to form our products, which are now CO2, carbon dioxide, and H2O, water. And you might already know that this type of reaction is an example of a combustion reaction. And we can see the new bonds that are being formed here. So some single bonds there, and here, as indicated, we have the formation of double covalent bonds, which you are now familiar with. All right, so that brings us to chemical equations. And if we look down the bottom, firstly, this is an example of a chemical equation. So it's simply an expression that uses chemical formulas such as CH4 and O2 and so on, and other symbols such as this arrow. You know that that's a reaction arrow to illustrate what reactants constitute the starting materials and what products are formed as a result of that chemical reaction. Reactants are always written to the left of the reaction arrow, and the products you can see are written to the right. Now, what you might not have seen in previous lectures is the use of these coefficients. So this number that's placed before the actual molecule itself, so number 2 there, and there's another coefficient there. So a coefficient simply shows the number of molecules of a given element or a given compound or a given molecule that react or are formed as products. So let's just make this clear. So the 2 that's placed there as a coefficient means that there are 2 molecules of oxygen that are part of these reactants. In other words, O2 plus O2. Same with this one, 2 molecules of water, so H2O plus H2O. Okay, so don't confuse those coefficients with these little subscripts that we see here, which indicates the number, how many of those atoms are in the molecule. All right, and that brings us to one of our laws of nature, I like to call them. Laws of nature, they're very important within chemistry and within other disciplines such as physics and so on. So this particular law is the law of conservation of mass. The law of conservation of mass states that in any enclosed system, That is a system that is closed to transfers of matter and transfers of energy. Within that closed system, atoms cannot be created or destroyed in a chemical reaction. They can only be rearranged. So that's the law of conservation of mass. And due to this particular law, we need to make sure that chemical reactions are balanced. And we use coefficients such as this number 2 to balance a chemical equation. A balanced chemical equation will have the same number of atoms of each element on both sides of the equation. It's that simple. So if we look at this example again with methane and oxygen, we can see that in the reactants, so on this side of the equation, there is 1 carbon, 4 hydrogens and 4 oxygen atoms. If we look at the atoms in the products, there is also 1 carbon, 4 hydrogens and 4 oxygens. So that is termed a balanced equation. And this is something that you should learn how to do. We'll look at an example in just a minute. So just some symbols that you might come across in chemical equations. So you already know that this is a reaction arrow. You might come across this, which sometimes indicates heat, but sometimes it indicates other things, and we'll come to that as we get there in lectures. All right, you'll see in your tutorial questions, or at least in a couple of them, you'll see the subscript S in brackets, which simply means a solid, L, liquid, G for gas, and AQ for aqueous solution. Okay, don't take too much notice of them, but at least when you come across them, you'll know what they're referring to. All right, so let's look at an example of how you go about balancing a chemical equation. Example, write a balanced chemical equation for the reaction of propane, which is C3H8, with oxygen, O2 to form carbon dioxide, CO2 and water, H2O. So firstly, write out the chemical equation using the correct formulas, which have just been given to you. So we've got propane written here as one of the reactants. Oxygen is a reactant, and then the products that are formed, carbon dioxide and water. Keep in mind that you can never, when you're balancing any equation, do not change ever these subscripts here. Because if you do, you'll actually change the identity of a particular compound or a molecule. All right, balance the equation using coefficients and tackle one element at a time. So let's start here. Let's balance first the carbons. All right, so we'll start on the left here. We've got 3 carbons on this side of the equation. On the other side, we only had 1. All right, so to balance those carbons, we place a 3. Use the coefficient 3 to balance the carbons. All right, so now they're balanced. Next, we'll look at the hydrogens. So we've got 8 there. On the right, how many do we have? We've only got 2. Okay, so let's look at what we do. Once again, 8 there, but we only had 2 on the right, so we place the coefficient 4 there in front of water. 4 times 2 is 8, so we now have 8 hydrogens there to balance those ones in the reactants. All right, what have we got left? We've got oxygen left, so we've got 2 here. On the other side of the equation, we have what looks like 1, but don't forget the coefficient. 4 times 1 is 4, so we've got 4 oxygens there, but only 2 there. So what do we do with that? Okay, here we go, 4 oxygens there. We only had 2 there, so we place a 5 there, a 5 there to balance the oxygen. Sorry, let me go back because I've missed a step. All right, sorry. So we had 4 oxygens here, but we also had oxygen here. How many do we have here? 2 by 3 is 6. So we have a total of 10 oxygens in the products, and we need to balance that with the 2 oxygens there. Okay, so in order to do that balancing, 10 oxygens on the right, as we just showed, but here we only had 2, so we use that coefficient 5. 5 2s are 10, so we now have the oxygens balanced on both sides of the equation. Okay, so it's as simple as that. It sometimes takes a little bit to work it out, but you'll get to practice that with a couple of your tutorial questions. All right, so that's a good one to learn, how to balance a chemical equation. All right, now let's move on to probably one of the most difficult concepts in chemistry. Not that it is a difficult concept, it is actually a really simple concept, but it just seems to be very difficult for many people to get their head around. And I think that's because it's the scale, the sheer scale of the number that we're trying to visualise, if you like. All right, so the concept here involves what's called the mole. But before we look at the mole, will you always, okay, we have a question, will you always add to balance? Yes, you always add to balance, that is correct. All right, so now let's look at a dozen. What do we mean by this quantity, a dozen? Oh, isn't that wonderful? Dozen. 12. 12 of something means 12 of anything, doesn't it? Yeah. What about a baker's dozen? What would a baker's dozen indicate? 13. Yes. Absolutely right. So a mole is exactly the same. It's exactly the same. It's just a quantity that's indicating a certain number. That's all it is. But that number happens to be enormous. It's 6.02 with a lot of numbers after that, 6.02 by 10 to the 23. Enormous. And that number is just called Avogadro's number, after the chap who actually came up with this number. So if you try to think of it like that, a mole is just a quantity that indicates a certain number of items. That's all it is. So you could have a mole of balloons, and you could have a mole of elephants. Yeah, say the same number of items, but they have very different weights, don't they? Very, very different weights. So keep that in mind. It's just like saying a dozen eggs compared to a dozen elephants. Yeah? Same number, different weights. All right, so we can use a mole, the term mole, in much the same way. We can refer to a mole of carbon atoms, a mole of carbon dioxide molecules, a mole of water molecules. We can say a mole of anything really. So just very briefly, where does this term mole actually come from? And keep in mind that when we're using the term mole with numbers, we usually use this abbreviation, MOL, rather than MOLE. So a mole was derived, or it's really defined as simply being the number of items, in this case, say the number of atoms, for example, found in 12 grams of pure carbon 12. 12 grams of pure carbon 12. That's how it was first set, basically as a reference, as a point of reference. Yeah. So that's how it was first devised, if you like. All right, so that term, the mole, can be used as a conversion factor to relate the number of moles of a substance to the number of atoms or molecules. But you don't have to worry about that, because this really is not relevant for us in the life sciences. So that's there just for your interest. All right, and so we'll move on to something that is very relevant and very important. And this is called the formula weight. Formula weight is often referred to as molecular weight, formula mass, molecular mass, all those terms really refer to the same thing. So the formula weight is simply the sum of the atomic weights or the atomic masses of all the atoms in a compound. And it's always reported in atomic mass units, in other words, AMU, which you might remember from lecture 2. All right, so let's look at how to calculate the formula weight of a compound. So calculate the formula weight for this compound here. Write out the correct formula, which we already have in front of us, and determine the number of atoms of each element from the subscripts. So if we look at this particular compound, it contains 1 iron atom, 1 sulfur, and 4 oxygen atoms. So once you've established that, it is simply a matter of referring to your periodic table of the elements. This is where you can always work out your formula weight for any given compound. And this is something that you should be able to do. You need to know how to do this. All right, people are dropping out and coming in. I hope that's not indicating poor bandwidth. All right, so step 2, multiply the number of atoms of each element by the atomic weight or the atomic mass. Remember, you get that from the periodic table. So if you look up iron, you will find the number 55.85 AMU. We've only got 1 of them. We've got 1 sulfur in the compound. You'll find this number on the periodic table, 32.07. And then we've got 4 oxygens. If you look up the atomic weight of oxygen, you will find that it is 16 AMU. We've got 4 of them for a total of 64. So the total formula weight that we have here for this compound is 151.92 AMU. All right, quite straightforward if you had the periodic table in front of you. So that's formula weight. And you'll actually see, on any container of any chemical, you will see something like this, FW with a number. Okay, so that's referring to the formula weight of that particular compound, that particular chemical. All right, get rid of that. All right, so that brings us to another important term. This term is referred to as the molar mass. So now you know what a mole is. You can imagine that a molar mass, the molar mass of a given compound or a given element is simply the mass or the weight of 1 mole of any substance reported in grams. And it just so happens that this number here that we were using to actually work out the formula weight is exactly the same number that we would use for molar mass, except that we use different units. So if we were to look at carbon on the periodic table and you wanted to see what the atomic mass or the atomic weight was, you would simply look at this number, 12.01 AMU. But if you needed to work out the molar mass of carbon, which is the mass of 1 mole, it's exactly the same number. Yeah, 12.01 grams in 1 mole of carbon. 12.01 grams in 1 mole. And of course, that refers back to our definition of a mole, how it first came about. All right, so keep that in mind when you're asked to calculate the atomic weight or the formula weight, if it's an actual compound itself, and the molar mass. The numbers will be exactly the same, but the units are different. Okay. All right, so hopefully that's straightforward for everyone. All right, now the molar mass can relate, as you probably imagine, can relate the number of moles of a substance to the number of grams. So the molar mass itself can be used as a conversion factor. So this is here just for your interest. So relating grams to moles. And we can also use the molar mass to relate the number of atoms or molecules. Once again, just for your interest. And the reason for that is because there are two very, very useful formulas, and there's probably not that many useful formulas in chemistry that relate to the life sciences that you're currently studying. But this formula here and on the next slide are very useful. And it basically allows you to convert back and forth when you need to. So the first one is shown here. M equals N times big M. So let's look at what this all stands for and how we can use this. All right, so this particular formula, I like to call the molar mass formula, and that's because it incorporates the molar mass, this big M here. So this molar mass formula allows us to link the mass of a substance, this little m, in grams to this little n, which is quite confusing because this little n refers to amount, but amount in moles. So one way of remembering this is when you see that little n, just think of number of moles rather than trying to remember that this means amount, but that amount has to be in moles. So mass in grams, number of moles, and this big M is molar mass. Okay, so that's the molar mass formula. Let's look at an example of how we can use that to solve a problem. So calculate the mass of glucose. So we're trying to solve for that there. Here's the chemical formula for glucose. Calculate the mass of glucose in a solution which contains this many moles of glucose. All right, so our unknown is mass. That's what we're being asked for. We're given the number of moles, little n, that is 6 × 10⁻³ mole. What about molar mass? We weren't given molar mass, but we were given the formula for glucose. And as I said earlier, whenever you have to work out the molar mass, you simply go to the periodic table and you look at the atomic weight of carbon times 6, the atomic weight of hydrogen times 12, the atomic weight of oxygen times 6, and add them all together. And if you do that, you should come up with the molar mass of 180 grams per mole. Remember that these are the units, always for molar mass. Okay, so next step for any formula is to cancel out any like units. So that means we can cancel mole with mole. And what does that leave us with? It leaves us with grams, the unit of grams. What are we trying to solve for? We're trying to solve for mass in grams, so you know you're on the right track. And if you solve that, you should get 1.08 grams of glucose. All right, now trying to figure out or trying to enter that type of number into a calculator, a lot of students are not familiar with how to do that. So if that's the case, then remember you will need to convert this back to a standard number before you work something like this out on the calculator. All right, now also keep in mind that when you're using this type of equation, you might also have to rearrange it. So let's make sure that you remember how to rearrange an equation. Now we're going to use the same method that we used for C1V1 equals C2V2. Oh, by the way, has anyone used that particular formula in any other subjects yet? C1V1 equals C2V2. Any other subjects? I know you have. I'm just waiting for someone to remember. Oh, physics. Okay, very good. That's, yeah, not quite what I was looking for, but that's a fair answer. Very good. No other subject? What about winemaking 1 students? Haven't you already considered that formula, C1V1? No? Okay, fair enough. All right, so let's say using this equation that this was the unknown. So let's say that was the unknown. Can someone tell me how I would go about rearranging this equation so that we can get this unknown by itself? Would anyone tell me what to do? All right, we've got an answer. Divide both sides by big M. You can certainly do that. All right, so we want to get rid of this one from that side of the equation. So we put it there, and cross those ones out. And that leaves you with N, number of mole, equals mass divided by molar mass. Good. Excellent. Let me clear that and get rid of that. All right, our second very, very useful formula. And this one is N equals CV. N equals CV, I call this the molarity formula. And so what do we mean by molarity? Molarity is simply a measure of solution concentration. So we're all familiar with that term concentration. So percentage can be a concentration. Volume per volume, weight per volume can be a concentration. Molarity is just another type of concentration. All right, so let's firstly look at the formula. N, what is N? You already know that that means amount in mole, so we'll simply say number of mole, equals concentration multiplied by volume. All right, volume is pretty straightforward, but keep in mind that the volume has to be in litres. So if you're given a volume that is in millilitres, then you'll need to convert that to litres. All right, so this little c refers to concentration, but this particular type of concentration refers to molarity. The term molarity, we normally abbreviate that to molar. And what does that actually mean? It means the number of moles per litre. So that's what molarity is referring to, moles per litre. Just to make things confusing, the unit that we normally use when we're talking about molarity is shown here, a big M. So reading this would read 0.24 molar solution. 0.24 molar solution. So don't confuse this molar with the molar mass, the big M that we used in the previous formula for molar mass. I know, a little bit confusing, but you will get your head around it. All right, so let's look at an example. That's the best way to really understand this. Calculate the amount of glucose. So amount of glucose, we're referring to N, number of mole, amount. In 25 mil, we already know we have to convert that because our volume has to be in litres, of a 0.24 molar solution. Okay, so that molar solution, that means we've already been given concentration. So that concentration is referring to molarity. All right, so the answer, we don't need to rearrange the equation at all. So number of moles, N, equals concentration, which is 0.42 moles per litre. Okay, so these are the units for this molar. Here, multiplied by volume. So we've converted our 25 mil to 0.025 litres. And the answer that you should get is 0.06 mole of glucose. All right, once again, we may have to or you will have to, I should say, rearrange the equation in some cases. So N equals CV, N equals C times V. So our molarity formula, you might have to solve for this one, concentration. Okay, once again, to rearrange that, divide both sides by V, cancel, cancel, and that leaves us with C equals N over V. Okay. All right, hopefully everyone's semi-comfortable with that, and hopefully you'll be more comfortable with it once you tackle those tutorial questions. Oh, and before we move on to the next concept, you'll also see that often you have to use both this molarity formula and the molar mass formula. You have to use one after the other in order to solve a particular question, which makes it even more tricky, knowing which one you have to use first before you can use the second one to solve a particular equation. But I have shown you some worked examples in your tutorial questions, so use those as a guide. All right, now we're going to look at one very important class of chemical reactions, very important class, and these are called redox reactions. The redox, the RED from redox comes from here, RED in the term reduction, and the OX from the word redox comes from here, oxidation. So two different types of reactions, oxidation reactions and reduction reactions, but they always go together, always go together. All right, so what is oxidation? Oxidation is the loss of electrons from an atom, whereas reduction is the gain of electrons by an atom. So this is very counterintuitive and very confusing because we're saying reduction, but then we say gain. So a really good way to remember this is to remember oil rig. So when you see any mention of oxidation reduction, otherwise known as redox, think oil rig, where oil stands for oxidation is loss, in other words, loss of electrons, and rig stands for reduction is gain, that is gain of electrons. Okay, so a really good one to remember, oil rig. And it's always great when I see students actually writing that on an exam, on their exam paper, oil rig. It's a great way to remember. All right, so both of these processes occur together in a single reaction called an oxidation reduction, or simply redox reaction. So this type of chemical reaction always has two components, one component that becomes oxidized and one component that is reduced. So a redox reaction involves the transfer of electrons from one element to another. All right, let's look at an example of a redox reaction. So we've got neutral zinc in this reaction, and we've got the copper cation that we can see here. So in this particular reaction, neutral zinc will lose 2 electrons to form the zinc cation, and the copper cation will gain those 2 electrons to form neutral copper. So we say because zinc has lost electrons, it is zinc becomes oxidized. The copper cation gains those 2 electrons, so copper is said to be reduced. All right, once again, remember oil rig. All right, and each of these two processes, the oxidation and the reduction side of it, can be written as what's referred to as a half reaction, individual half reactions. So let's look at the oxidation half reaction and look at how we write that. So the oxidation part of it is written as neutral zinc, which we can see up here, And because it loses 2 electrons, we write that loss of electrons here to the right side of the reaction arrow. So zinc becomes oxidized, that is, it now picks up a 2+ charge because it's lost 2 electrons, and that's written as +2 electrons. Okay, if we look at the reduction half reaction, that's simply written as the copper cation. Here it is here. Remember, it picks up the 2 electrons, and because it gains 2 electrons, it's said to be reduced. We write that gain of 2 electrons, as you can see here, to the left of the reaction arrow. And here's the product here, neutral copper. Okay, so that's what we refer to as half reactions, and you'll see some of them also in your tutorial questions. Okay, another concept here. Compound, also a very confusing one. Another counterintuitive concept. All right, a compound that is oxidized while causing another compound to be reduced is called a reducing agent. Okay, so zinc acts as a reducing agent. Even though zinc itself is oxidized, it acts as a reducing agent because it causes the copper cation to be reduced. Then we can look at a compound that is reduced while causing another compound to be oxidized, and that compound is called an oxidizing agent. So the copper cation that we can see up here, even though it's being reduced itself, it's causing zinc to be oxidized. Yeah. So we call the copper cation an oxidizing agent. Once again, very confusing until you really get your head around it. And I know it's very confusing because this is often part of a test or exam where students get very confused. So a really good one to try and get your head around. All right, so the burning question, how do you know what's going to be oxidized and what's going to be reduced? Okay, no, there's no easy answer to that, but there are some general guidelines that we can follow. So when you're working through some of your tutorial questions, remember slide 29. This will give you some really useful tips here. All right, so let's start here with metals. Metals group 1A, group 2A metals. These tend to undergo oxidation reactions. That is, they tend to lose electrons. You can see here, they've picked up charges because they have lost 1 or 2 electrons. Okay. All right, what do we have here? Group 7A, group 6A anions. Yeah, they also tend to be oxidized. As you can see here, loss of 2 electrons because we started with 2 chlorides, loss of 4 electrons because we started with 2 oxides. Okay, now I want you to note here the balancing that's going on. Okay, the balancing is indicated here by the use of these coefficients. So keep that in mind. 2 chlorides. Here we no longer have chloride, we have chlorine, and the 2 is indicated now by a subscript. Okay, whereas here was indicated by the coefficient. All right, so that's some good guidelines for oxidation reactions. Let's look at reduction reactions. All right, neutral halogens, group 7A and group 6A elements, these tend to be reduced. And we can see the gain of electrons here, the gain of electrons there to form anions. And then the same applies here. So when we're looking at cations, these also tend to be reduced. We can see the gain of electrons here to form these neutral elements. All right, so some very good guidelines there. But as I said, it's not actually that straightforward to work out what will be oxidised and what will be reduced. It really depends on the two active interacting elements. All right, now here's just a classic example of a redox reaction, and it involves the rusting of iron. Everyone's probably familiar with rusty old car, for example. I've had many of them in the day. All right, so let's look at this. So here we have some pure iron atoms before undergoing that process of rusting. And this is what we start with, 4 iron atoms, neutral iron atoms. Now, what happens to pure iron in the presence of water? In the presence of water or water vapour, the iron atoms will interact with oxygen. They'll interact with oxygen to form iron oxide. And to understand what that means, now look at what we've got here. So we've got now charged iron atoms, and we've also got the red oxides. And the problem with this iron oxide is that the interactions between these atoms here are nowhere near as strong as the interactions between these pure iron atoms, the neutral atoms. Okay, nowhere near as strong. So now this material here, this iron oxide, becomes very, very porous, very permeable to further entry of water and oxygen. And that's why once rust starts, it's very, very hard to stop it. In fact, it's almost impossible to stop that occurring. All right, so that's the process of rusting. And of course, that's why when rust starts, it becomes very flaky and it starts to crumble because of that increased permeability to water and oxygen. All right, and our final slide here. Here we look at different ways in which substances can be oxidised or reduced. So before we said that oxidation and reduction involves the loss of electrons and gain of electrons, but oxidation can also result from the gain of oxygen, the gain of oxygen atoms, that's also referred to as oxidation. And of course for those students that are studying winemaking, you're probably already familiar with that concept of oxidation of wine when it's exposed to too much oxygen. So, and oxygen can also result from the loss of hydrogen atoms. And we'll look at an example here. And of course, reduction can result from loss of oxygen, so the opposite to oxidation, and gain of hydrogen. All right, and we go back to our example of methane and oxygen reacting in a combustion reaction to form carbon dioxide and water. All right, so in this particular combustion reaction, so our methane CH4 is oxidised to carbon dioxide, CO2. So in that process, it has gained oxygen and it's lost hydrogen. So methane is said to be oxidised. Now, if we look at the oxygen here, oxygen is said to be reduced in this combustion reaction. Here it is. It's actually gained hydrogen. So gain of hydrogen is reduction. All right, and that completes this lecture. How's everyone feeling so far? Okay, everyone's gone a bit quiet. It's gone very... Fine, one glass at a time. That's what I like to see, Rob. Mostly okay, good. All right. Okay, I know it's a bit much, but it will be, someone needs a refill. Yes, it will get better when you have a look at some questions. All right, now, before a break, we're going to make a start on lecture 6, biological molecules. In this lecture, really we start to enter the realm of biochemistry. So this is a discipline where, you know, as the name implies, chemistry meets biology. Very important for all of us in the life sciences. All right, and we're going to take a look at obviously that link between the chemistry and the biology, and we're going to do that by looking at the 4 major groups of biological macromolecules. 4 major groups. Can anyone tell me what they are? What are the 4 major classes of biological large molecules? There's only 4 of them, and they're essential. Any ideas? You might have an idea if you've already maybe had a quick look at this lecture. 4 major classes of biological macromolecules. No takers? Organic. All right, okay, that's all right. I'll start you off. Carbohydrates. Carbohydrates, that's all right. Good try. Good try. That's fine. Good to have a go. Carbohydrates. Here we go. Now we've got fats. Yes. Let's say lipids. Lipids, which includes fats. Yep. And we've got another one. Yes. Proteins is another one. There's one more. Proteins. Carbohydrates, lipids, proteins. And the last one is a little bit tricky. Amino acids go with proteins. Enzymes go with proteins. Alcohol. I like that one, alcohol. And minerals and vitamins. Yeah. Yes, they fit into one of these classes. Yeah. I like the alcohol. All right, so the 4th one, I nearly forgot myself. The 4th class of biological macromolecules, the nucleic acids. Now, you might not be familiar with this one, but you will be familiar with the 2 major types of nucleic acids, which are DNA. Everyone's heard of DNA. Yep. And the other major type of nucleic acid is RNA, which you may not have heard of. All right, so we're going to focus on those 4 for probably another 10 minutes, and then we'll have a break. All right. Time for a refill in 10 minutes, Rob. All right. Organic molecules. We're not going to go into organic molecules tonight. We will spend a lot of time on organic chemistry in the 2nd semester. But organic molecules are basically molecules that contain carbon. That's a very good basic definition of organic molecules. Biological molecules, biological macromolecules, composed principally of the atoms of these 6 elements. Okay, nearly always carbon. I always have to remember to say nearly because there's always exceptions. Nearly always carbon and a lot of hydrogen, and then many examples where there's oxygen, and then there's examples with nitrogen and phosphorus and sulfur. Carbon is a big one. It features very, very heavily in organic molecules, in biological macromolecules, because carbon atoms, as you know, have the ability to form 4 covalent bonds with other atoms. So carbon-laden molecules can form straight chains, they can form branches, they can form rings, they can form any manner of different types of molecules. That's why carbon is often referred to as the party animal on the periodic table of the elements. All right, carbon, as I said, hydrogen also features very heavily in biological macromolecules. And as you probably can imagine, hydrogen tends to bond quite readily with carbon. And those covalent bonds between carbons and hydrogens are energy rich. They're energy rich bonds, and that energy is extracted in processes that we won't talk about this semester, but we'll certainly go into them next semester. Really important metabolic processes. All right, so there's over 15,000 different unit organic molecules that have been described and characterised in living organisms, but only about 29 are needed to describe most of the basic biological molecules. And we're going to look at a few of these in more detail in this lecture. All right, so biological macromolecules such as proteins and carbohydrates and so on. They're large molecules and they're composed of repeating units. And those repeating units are assembled by a particular type of reaction that belongs to this broad class of reactions called condensation reactions. The specific type of condensation reaction which allows these repeating units to join together to form these large molecules are called dehydration reactions. So dehydration reaction is a condensation reaction where the small unit molecules, which we call monomers, you can think of them as the building blocks that make up this large macromolecule. So these unit molecules or monomers react together to form larger molecules, which we refer to as polymers. All right, here are some examples of these unit organic molecules that are really, really common and super abundant in organisms, regardless of whether you're studying plants or animals. The same thing applies. All right, glucose, triacylglycerols, phospholipids, amino acids, and nitrogenous bases. So, of course, glucose is an example of which class does it belong to? I should have left the 4 classes up. Glucose belongs to what class? Carbohydrates. Good. It's a sugar, belongs to the class of carbohydrates. Any idea about this one? Triacylglycerols and also phospholipids. What class of biological macromolecules? Yes, lipids. These are both examples of lipids. What about this one? Amino acids. What class do the amino acids belong to? Proteins, correct. They make up proteins. And what about nitrogenous bases? What do they make up? Nitrogenous bases. You probably won't know this one unless you've studied some biochemistry or even biology, if you've studied biology in BCE. All right, nitrogenous bases basically make up or are part of the monomers that make up DNA and RNA, which are our nucleic acids. All right, so let's start with the all-important carbohydrates. Not a problem. Not a problem. I understand. Everyone's home with the kids at the moment. Okay, so carbohydrates are the most abundant organic compound in living organisms. Here is the general formula for a carbohydrate. So for every 1 carbon and 1 oxygen, there are twice as many hydrogens. And to this little n here to the outside of the brackets simply means any number of these molecules. Any number of those molecules can be actually joined together, bonded together to form various types of carbohydrates. All right, the function of carbohydrates. Mostly they function as energy storage molecules or as structural molecules. Okay, they're the 2 major ones, well, the 2 major ones that we'll be looking at tonight. But carbohydrates also play an important role in the interaction between cells, whether these are cells within the body of a plant or in the body of an animal. And there you'll also see later on that nucleic acids, so remember DNA and RNA, there are carbohydrates that are important building units of nucleic acids. All right, so 3 very broad groups of carbohydrates, the monosaccharides, the disaccharides, and the polysaccharides. So let's look at the monos. Here we have monosaccharides. These are the simplest of the carbohydrates. They consist of a single molecule, that is a single unit, a single monomer, you can say. They are classified according to the number of carbon atoms they contain. And those that contain 5 or more carbons tend to exist mostly in ring-like structures, cyclic structures, most of the time, not always. Okay, the monosaccharides are very soluble in water and they have a varying degree of sweetness depending on the monosaccharide. All right, let's look at an example. Well, we have actually 3 examples here. So to the right, we have glucose, galactose, and fructose. And you can see here how they form these cyclic structures. Well, most of the time anyway. All right, we're not going to focus on the cyclic structure. We're going to focus here on this linear structure. And I'll just get my pen here. So let's just look at this one here. So this is glucose. Don't worry about that D. This is glucose here. And we can see that 1 glucose monosaccharide, 1 molecule of glucose contains 6 carbons. Each of those carbons are covalently bonded to either hydrogens or to oxygens, which in turn are bonded to an oxygen, to a hydrogen rather. So keep in mind when you see that OH there, so that's the C, and that's the O, and that's the H. So basically that bond has been left out and it's just written as OH. So we've got carbon, hydrogen, and oxygen. That's it. That's all that you need to make glucose. And of course you can see the presence of 1 double covalent bond there. All right, let's look at this example here. Another very abundant monosaccharide. This one is galactose, and it's almost identical. Can someone tell me what the difference is between glucose and galactose? Who can spot the difference there? Very subtle difference between the 2 of them. All right, you're on to it. 4th line. 4th line, 2nd, HO. Yes, that's it. You've got it. You've got it. So if you look here, here we go. So here, so on this carbon, the OH is to the right, but on this carbon in galactose, the OH is to the left. That's the only difference. And while that sounds like it's no difference at all, it actually does make quite a big difference, but we won't go into that here. All right, what about this one? Fructose. Also very common monosaccharide. The difference here is very obvious, because we can see that instead of the carbon-oxygen double bond there, it's not on that first carbon, it's here on the second carbon. Okay, quite different, in fact. All right, so that's our monosaccharides. Let me clear that. And I think it's time for a refill of that glass. What do you think? Time for a break. Oh, sounds good. Yes. All right, let's break. What's the time? 7:34. Let's break for 10 minutes. I'll see you at back here, 7:44. Okay? Oh, 7:44. Yes, you certainly can ask questions. I think he's gone for a refill. He's not answering. Yep, he needs another bottle. Not just refilling the glass. We're on to him. Has anyone seen the videos? The winemaking one videos? No? No one. Oh, you've seen them? Not too bad. Oh, rubbish. Are you saying the wine's rubbish or the videos were rubbish? Oh, these are just a couple of practice videos that were taken in the winery on Friday, and they're up on the winemaking one Moodle site. So it sounds like someone doesn't like them. Not at home. They look good. Thank you. Thank you. I've got all the agronomy ones ready to go too. I've got them all up, but you can't see them yet because I had to just check all the links, but they'll be available by tomorrow. Oh, not the videos are not rubbish, just your drink choices. Oh, didn't you have anything good in the cellar? All right. Oh, it's time. Sorry. Oh, let's get back to work. All right. So we've looked at monosaccharides. Now our second broad class of carbohydrates are the disaccharides. As the name implies, di, that means two monosaccharides are joined together. So monosaccharides may link together in a variety of different ways, and that link or that bond that joins them together is called a glycosidic linkage. And when two monosaccharides link together via a glycosidic linkage, they will form disaccharides. And we've got some examples here for you. So let's look down here. So sucrose, everyone's familiar with sucrose. Sucrose is a disaccharide which is formed from one glucose and one fructose monosaccharide. And remember that linkage that keeps them together, that bonds them together, is called a glycosidic linkage. We won't go into details about that here. All right, another example, lactose, which is formed from one galactose and one glucose. And here we've got another disaccharide, maltose, which is two glucose monomers bonded together. All right, so sucrose. No, let's go back here. All right, sucrose. So sucrose tends to be the major transport carbohydrate in plants. The major type of carbohydrate that's transported through plants. Can anyone hazard a guess as to why this is the case? Why would plants not just transport glucose? Why wouldn't they transport glucose from one part of the plant to the other, from where it's made to where it's needed, for example? Why not glucose? Why sucrose? Any idea? It's not really obvious. Why do plants transport sucrose and not something like glucose or fructose? Okay, the reason for that is that glucose, for example, tends to be quite reactive. It's quite reactive. So it's very, very readily metabolized as it moves around, but sucrose, not so much. So sucrose can be transported from one part of the plant all the way to a very distant part of the plant without undergoing metabolism along the way. So that's the main reason for that. All right, now let's move on to the third class of carbohydrates, and these are the polysaccharides. These are very large carbohydrates, or can be very, very large. They may consist of chains of hundreds or even thousands of monosaccharides linked together. They form two major functions. One, they are structural components, and two, they function as storage reserves. So as structural components, we tend to see polysaccharides in the cell walls of plants, very much so in the cell walls of fungi, and even in the cell walls of bacteria. Well, that is those bacteria that have cell walls. And then also in association with certain proteins, we find polysaccharides in connective tissue, in animals, and also in the exoskeletons of animals. Polysaccharides also function as storage reserves in all organisms. And when we're talking about storage reserves, we're talking in particular about storage of energy. All right, our first example is that of a structural polysaccharide, and of course cellulose would have to be number one because cellulose is the most abundant polysaccharide in the biosphere. So we find cellulose predominantly in plants. So for example, in this tree trunk, that would consist of about 50% cellulose. Even more so in cotton, which is about 90% cellulose. But we can also find cellulose in some unexpected places. So some bacteria, so this is a bacterial cell, they'll form these protective layers around the cell. And those protective layers can also contain cellulose, depending on the species of bacteria. All right, let's look at the chemical structure of cellulose, this structural polysaccharide. So here we have one glucose monomer bonded to another glucose and another one and another one. All right, so cellulose is made of glucose monomers, many glucose monomers all linked together. Here's another view. This is a surface view of glucose monomers and the way that they're joined together. A little bit hard to see. So there's one there linked to another one there and there and there. So in a straight chain, and then they are linked to another chain here of glucose and so on to form a sheet of cellulose. And then if we take a side view of that sheet, here's the side view here. So here's the side view of one sheet, that joins to another sheet, and so on until you actually have a fibre of cellulose. And that's what we're seeing here. These are actual fibres of cellulose running in one direction, and then you can see them underneath also running in the opposite direction. And this is an actual image of a plant cell wall and how those cellulose fibres are organised. All right, here's another example of a structural polysaccharide, and this one is called chitin. Chitin is very similar to cellulose. It has the same ability as cellulose to associate laterally to form those fibres which we saw. Those fibres are called microfibrils. We find chitin predominantly as structural components of the walls, the cell walls of fungi, also in the exoskeleton of arthropods. Chitin is really tough, very, very tough compound. So if you ever try, or you need to try, to break open fungal cell walls, you'll have a lot of trouble. Bacterial cell walls are easy to break open, but not so much fungal cell walls because of this this polysaccharide. All right, so chitin chemically looks like this. So this is very similar to glucose, but it's not glucose, and it's attached, as we can see, to other monomers here. And the difference between glucose and this particular monomer is the presence here of a nitrogen-containing group. Okay, that's the difference there. So these can be looked at as glucose derivatives, if you like, but they're certainly not glucose monomers. All right, so that's chitin, and that's our structural polysaccharides. Now let's look at storage polysaccharides. So ultimately, we know that all organisms on Earth depend on photosynthesizing plants and also photosynthesizing algae, and to a much lesser extent, photosynthesizing bacteria, to convert the energy from the sun into the chemical energy of carbohydrates. So they are our source, our ultimate source of carbohydrates on the planet. Okay, storage polysaccharides, the most important, or probably the most abundant. One is starch, and that's for plants, and for animals, we can consider this one glycogen. So starch tends to be found as a storage polysaccharide, energy storage polysaccharide, as granules in certain parts of plants. So a chloroplast, you don't have to remember all these different plant parts here, but if you're interested, they're found predominantly, but not exclusively, in plant chloroplasts. That's where photosynthesis occurs, and as transient storage material, and also in tubers, in seeds, in young cotyledons, and so on. So animals, of course, store carbohydrates in their tissues, in certain tissues, as glycogen. Now, one thing to note is that starch is made of glucose. Once again, glucose monomers joined together, just like cellulose. Glycogen is also made of glucose monomers joined together, but in a different way to starch and in a different way to cellulose. Glycogen is also a little bit different in that those glucose monomers are all bound to a central protein, a core protein. So a little bit different, but they're basically both made up of glucose monomers. All right, and here we have some starch granules in plant cells. And you can see that clearly, that starch granule there, in a chloroplast and in a potato tuber. Some great images there. All right, moving away from carbohydrates, now let's look at lipids. The lipids are a really diverse group, so diverse that we can't just name one or a few different unit organic molecules that characterises all the different lipids. They are an incredibly diverse group. The group of lipids, this group known as lipids, includes fats, oils, waxes, phospholipids, steroids, for example, cholesterol, and carotenoid pigments. So quite a list already, and that's just a few of them. How do we define a lipid? Lipid can be defined as a substance that can be extracted from biological materials using non-polar organic solvents, but you can't extract them just using water. Okay, So there's a few terms here that might be foreign to you. Don't focus on that, okay? This is simply a definition. All right, lipids are composed principally of carbon, hydrogen, and oxygen. So exactly like carbohydrates, but they differ from the carbohydrates in that they have a much lower proportion of oxygen. All right, here are some of the diverse functions of lipids in biological systems. They function in storage, protection, structural components, signaling molecules, pigments, photoreceptors, cofactors in biological reactions. The list would go on and on if you've really researched this. Here are some different types: triacylglycerols, waxes, phospholipids, sterols, steroids, steroid hormones, glycolipids, isoprenoids, and so on, carotenoids in plants and in animals, and the vitamins that someone referred to earlier. Some of the vitamins, at least, are lipids. All right, you don't need to remember this. This is for your interest and to put it into some sort of context there. All right, we're going to start with some examples of simple lipids. And our first example involves what are called fatty acids. Fatty acids basically consist of a chain of carbons, which is referred to as a carbon backbone. And the length of the chain can vary depending on the fatty acid, can vary from 12 to 20 carbon atoms. Those fatty acid chains that contain only carbon-carbon single covalent bonds are referred to as saturated fatty acids. Other fatty acid chains that contain at least one carbon-carbon double covalent bond are referred to as unsaturated fatty acids. And here are some examples for you of two fatty acids. So this one on the left is called stearic acid. It has 18 carbons. And you can see that carbon backbone, that is that long chain of carbons. Each one is bonded to 2 hydrogens or 3 hydrogens, as in this one here. Stearic acid, this is a type of fatty acid that you would normally find in animal fats. So that's stearic acid. This one on the right, oleic acid, also has 18 carbons. There it is. So all almost identical, but this one is predominantly found in plant fats. What is the difference between these two? Who can spot the difference? There is one difference between stearic acid and oleic acid, and it makes an incredible difference. Yes, the double bond. All right, so remember on the previous slide we said that saturated fatty acids contain only carbon-carbon single covalent bonds, which is the case here. Unsaturated fatty acids, and oleic acid is an example, contain at least one double covalent bond, at least one. Okay, so there are our fatty acids, or rather a couple of examples of fatty acids. And now we move on to waxes. So waxes are totally insoluble in water due to the non-polar nature of these molecules. They occur as coatings on the surfaces of above ground plant parts, which acts very effectively to reduce water loss by evaporation. Waxes are also secreted from preen glands to coat the surfaces of feathers of aquatic birds, from epidermal glands of mammals to coat fur, and also are found being secreted by certain cells of insects. All right, our next group of lipids, a simple lipid still. These are the triacylglycerols, also known as triglycerides. They are referred to as neutral fats or oils. And these are stored in special storage cells of fat tissue otherwise known as adipose tissue in mammals, but also triacylglycerols are found in certain plants, particularly in certain seeds, castor bean, canola, coconut, oil palm, flax, and also in fruits such as avocado and olive. And here are some fat cells found in animals. They are called adipocytes. And you can see some very small fat cells here, or one there, and very large fat cells that are really, really swollen as they contain so much fat. All right, now let's move on to the complex lipids. Where are the complex lipids found? They form incredibly important components of cell membranes. So we're talking about cells that make up the body of a plant and make up the body of an animal. And each of those cells are surrounded by biological membranes, which are full, absolutely full of these complex lipids. Complex lipids are also found in this covering of nerve cells in animals called the myelin sheath. Complex lipids also contribute to what's called surfactant, and this is a substance that reduces surface tension in the lungs of an animal. Very important to keep lungs functioning. All right, complex lipids also include essential oils of plants and also molecules involved with photoreception. All right, let's look at this very important type of complex lipid. These are the phospholipids. So phospholipids, let's look at an example. Before reading all that, let's look at this example here, this illustration of one phospholipid molecule. And you can see that it's divided into two parts. So this part here is called the head, the phospholipid head, and these two parts down here are referred to as the tails of the phospholipid. All right, the phospholipid head is said to be hydrophilic, and that term hydrophilic means that it's water loving. It can associate with water. The tails of this phospholipid molecule, on the other hand, these two tails are referred to as being hydrophobic. And hydrophobic means water fearing or water hating. So these two tails do not like to associate with water. They chemically cannot associate with water. So we have this really interesting combination here within the one molecule of a hydrophobic region and a hydrophilic region. That is a region that likes to associate with water and one that doesn't like to associate with water. And molecules such as this phospholipid that have both of those properties are termed amphipathic. Okay, they have both the water-loving and the water-fearing region within the one molecule. Okay, so phospholipids have a non-polar region contributed by the hydrocarbon chains, which is what we see down here. And these two chains happen to be fatty acids, which we just learned about a couple of slides ago. All right, so those hydrophobic fatty acid chains are insoluble in water, that is, they're hydrophobic. And the phospholipid also has a hydrophilic region, and that is the water-soluble region, the hydrophilic head of the phospholipid. All right, so I'm going to skip this slide, but I'll come back to it in just a second. Let's look at this very common representation of a phospholipid with the head, hydrophilic head, and the two hydrophobic tails. Now let's look at what would happen if you could just have those phospholipids contact water. What would happen? Just look at this diagram here. And as you can imagine, those hydrophilic or water-loving heads would very happily associate with the water molecules here, but the hydrophobic or water-hating tails will try to escape from the water, try not to associate with the water. Now imagine if you could just agitate that solution and force those phospholipids into the actual solution itself. What do you think might happen? And now we're looking at this image here. So now the phospholipids have rearranged themselves into this self-enclosed sphere, this vesicle, if you like. And this is referred to as a mycele. And the way that the phospholipids have arranged themselves is, once again, so that those hydrophobic or water-loving heads are in contact with the water, but the hydrophobic or water-hating tails are all congregated to the inside of that little sphere, trying to escape contact with the water. All right, so that's myceles. Now let's look at biological membranes. So remember we said that that animals are made up of cells, plants are made up of cells, and all those cells are surrounded by a biological membrane. And those biological membranes just happen to be made up of 2 layers of these phospholipids. And we can see those 2 layers, once again, associating with each other to form a self-enclosed sphere, a vesicle. So they're enclosing the contents of the cell. And these 2 layers here are referred to as a lipid bilayer. So for this upper layer, phospholipids, once again, the heads, the hydrophilic heads, are associating with this aqueous medium outside the cell. This lower layer of phospholipids, the heads, are associated with the aqueous interior of the cell, but all those hydrophobic tails are all facing towards each other within the actual membrane itself and escaping from the water. So that's quite amazing how that occurs to form living cell membranes. So now we can return to the previous slide, and which will make more sense to you. So phospholipids in water may rearrange themselves to form a self-sealing closed vesicle, closed sphere, and the surface of that sphere is a lipid bilayer. The hydrophilic heads of the outer layer of that bilayer are in contact with the external aqueous phase, and the hydrophilic heads of the internal layer of the phospholipids are in contact with the internal aqueous phase. The interior of that lipid bilayer is formed by association of those hydrophobic or water-hating hydrocarbon chains. So phospholipid bilayers are the basic structural units of all biological membranes, all cells, all biological membranes, regardless of whether we're talking about animals or plants or even Saccharomyces cerevisiae. All right, moving on now to our next group of biological macromolecules, and these are the proteins. Everyone's familiar with proteins, the building blocks, the monomer units, if you like, are the amino acids. So all proteins are made from amino acids. The amino acids are joined together by special type of bonds called peptide bonds. And when they all join together, they will form a polypeptide chain. We don't call it a protein yet. It has to go through several steps before it becomes a functional protein. At this stage, when the amino acids are just linked together in a chain, we refer to it as a polypeptide, which of course is a polymer. And the monomer units are the amino acids. All right, the component amino acids are selected from a standard set of 20. And this is in both plants and animals. Let's look at the general structure right here of an amino acid. Okay, quite simple really when you look at them. All amino acids have this core carbon called the alpha carbon. And we know that carbon forms 4 covalent bonds. All right, one of those covalent bonds is to a hydrogen atom. The second covalent bond, this one here, that's bonded to a nitrogen, which in turn is bonded to 2 hydrogens. If we put them all together, this is referred to as an amino group, NH2. Let's look at this bond here. It's bonded to a carbon, which in turn is bonded to an OH, and a double covalent bond to an oxygen there. Put them all together, COOH, and that's referred to as a carboxyl group. All right, here's the remaining covalent bond from that alpha carbon, and that is bonded to a something different depending on which amino acid we're looking at from that standard set of 20. So this is where they all vary. They all vary at this point, and this is called a side chain group or simply an R group. Okay, so once again, all amino acids have this exact structure, but they differ here at this side chain or R group. All right, proteins are classified in different ways. They can be classified according to their biological function, according to their composition, or according to solubility. Okay, we've got lots of examples here, and we'll look at some of them. You don't need to remember these. So classification according to biological function, they can be classified as enzymes, as structural proteins, contractile proteins, and so on. Classification according to composition, they can be classified as lipoproteins or glycoproteins, nucleoproteins, and so on. According to solubility, they could be water soluble, in which case they're classified as globular proteins. They could be membrane proteins or fibrous proteins. All right, and here we have the standard 20 amino acids on this slide and on the next slide. So let's look at the general structure once again. Let's look at this first one here, glycine, which happens to be the smallest of the amino acids. So let's look at that structure. Alpha carbon in the middle, 4 covalent bonds, hydrogen here, amino group here, carboxyl group here. Okay, they're all the same in that regard. Here's that variable side chain group or R group. In this case, the R group is simply a hydrogen. In the case of the amino acid alanine, the R group is a CH3, so still a very small amino acid. And now you can see they start becoming larger and more complex. All right, this particular group of amino acids here are referred to as the neutral non-polar amino acids. Let's look at what they're composed of. They are all composed of carbon, hydrogen, oxygen, nitrogen, and then in a couple of cases we have sulfur here and here, so in cysteine and methionine. Okay, so that's the first group of standard amino acids, and here are the rest. So in green, we have the neutral polar amino acids. In pink, we have the 2 acidic amino acids, and here we have our 3 basic amino acids. So once again, you don't have to remember these. If you were doing a major in biochemistry, it would be different. All right, so we said earlier that when amino acids join together, to form a chain of amino acids. That's referred to as a polypeptide. And the order in which those amino acids actually link together within that chain defines what's referred to as the primary structure, structure of the protein. And that's because the order of those amino acids will impart different biological properties to the protein once it's completed its formation. We haven't actually completed it just yet. All right, so that's the primary structure. Then we can talk about the secondary structure. And this is actually determined by the arrangement of those variable side chains, those variable R groups that you would find on either side of the peptide bonds that are actually bonding those amino acids together. And that arrangement of those different R groups along that chain of amino acids will actually determine different structural patterns or structural motifs that start to form within that, that newly formed protein, or what will become a protein. Two of the common patterns or motifs, the most common that are seen, are called the alpha helix and the beta sheet, or beta pleated sheet. And we can look at that here. All right, so once again, here is the primary structure. That is the order of amino acids that are linked together into a polypeptide chain. Okay, if we were to look at just this region here and here, we can see there's one part of the chain there. There's another part of the chain there, and we can see that they are actually interacting with each other. Those two chains are interacting with each other. And when they interact in this way with each other, it forms one of these patterns or one of these motifs, secondary structural motifs called a beta sheet. And it reminds me always of a piano accordion, that particular shape that it takes. And here's the shorthand symbol for a beta sheet. So an arrow in one direction and another arrow going in the other direction. All right, so that's the first of the common structural motifs. The second one is the alpha helix, and this is pretty straightforward to visualize. So we can see a chain of amino acids that have formed this alpha helical structure. Okay, and this is what it looks like in one of these illustrations. All right, not so clear in this primary structure here. All right, so secondary structure, think beta sheet or beta pleated sheet and alpha helix. Oh, and you can also see, you might be able to see the formation of these bonds here. They're not covalent bonds, different type of bond there. The red dots that you can see that are enabling this alpha helix formation to occur. All right, so that was the secondary structure of a protein. Now we could look at the tertiary structure. And the tertiary structure actually refers to the final folded three-dimensional shape of the protein due to side chain interactions between the side chains. So these are three representations of proteins here. This one consists primarily of the alpha helix. This one predominantly contains beta pleated sheets, and this one here is a combination of the two. All right, so that's the tertiary structure. We still don't have the final functional protein. For that, we need the quaternary structure. So at this level, we may have the association of two or more polypeptides together. They associate together, and that's when you have the final functional protein that can carry out its task. In this particular example here, we have 1, 2, 3, 4 polypeptides actually associated together to form this particular protein. 2 alpha polypeptides and 2 beta polypeptides. All right, and here we just have some different types of proteins, which you're probably familiar with. Everyone's heard of keratin. Just think of hair, nails, horses' hooves would be another good example. Silk, everyone's familiar with. Collagen, and here's another one, elastin. And some photos here, so you can imagine in the fur of a mammal, you would expect to find keratin, bird's feathers, tortoise shell, and a spider's web. All right, and now we come to our final group of biological background molecules, the nucleic acids. Nucleic acids, what are we talking about here? What are the 2 major types? We're talking about DNA and RNA. These are found in all living cells. All living cells contain DNA and RNA. All right, so now that you know that, can anyone tell me why are viruses not considered to be living organisms? Why are viruses not considered to be living organisms? Good answer. They only have RNA. You're on the right track. You are on the right track. Absolutely. Anyone else? Why are viruses, well, sorry, let me rephrase. What is one reason why viruses are not considered to be living organisms? Anyone else? Something to do with what I've just put up here. Someone's on the right track there. All living organisms contain DNA and RNA. No DNA. All right, the answer is viruses have either one or the other. They don't have both. They never have both. Virus only has DNA as its nucleic acid or RNA. But all living organisms have both in all their cells. Well, sorry, most of their cells. Okay, so very interesting in light of the current situation. Of course, I had to bring that up. All right, so back to nucleic acids. They are found in all living cells. DNA is the nucleic acid molecule in which information on cell structure and cell function is stored. And this information is transferred from one generation to the next. So an incredible molecule. DNA, very simple molecule, and yet it contains all that code, all that information that will determine whether the offspring is going to be a plant or an animal or a bacterium. That's absolutely amazing. And when you learn all about DNA, you'll see how amazing it is. All right, so what's the building block? What is the monomer of the nucleic acid? That is either DNA or RNA. The building block, the monomer unit, is the nucleotide. Nucleotide has 3 parts. It has a nitrogen-containing base or a nitrogenous base. It has a monosaccharide, a sugar. That sugar is either ribose or deoxyribose, depending on whether it's a nucleotide found in DNA or in RNA. The nucleotide also contains a phosphate group. All right, let's look at that in more detail on the next slide. All right, so once again, let's look at this general structure of a nucleotide, which makes up DNA and RNA. Here's the nitrogenous base, here's the monosaccharide, S for sugar, and P for phosphate. Altogether, that is one nucleotide. Now let's look at the chemical structure of a nucleotide. All right, here's the nitrogenous base, and you can see lots of nitrogen in there, single and covalent bonds, lots of carbon and a little bit of hydrogen. This particular base is called adenine, and there are actually 5 different bases. 4 of those 5 are found in nucleotides in DNA, and 4 of those 5 are found in nucleotides in RNA. All right, this one is adenine. All right, that nitrogenous base is bonded to a monosaccharide, a sugar, and this particular sugar is called ribose. And because it's ribose rather than 2-deoxyribose, we know already that this nucleotide is found in RNA. Ribose, RNA. If the sugar was 2-deoxyribose, then we would know that the nucleotide would be one of the nucleotides found in this nucleic acid, DNA. All right, so here we have the ribose sugar, and it is bonded here to a phosphate group, which thankfully doesn't change depending on whether it's RNA or DNA. That would make it really confusing. All right, so let me clear that and go on to the next slide. Now, so I said that there were 5 different types of bases and therefore 5 different types of nucleotides, and they give them, they put them into 2 different groups, those 5 nucleotides into 2 different groups, which are shown here, not particularly important for us. One group is called the pyrimidines, one group is called the purines. So the pyrimidines include these nucleotides, uracil, cytosine and thymine, and they're given the abbreviations of simply U, C and T. And the pyrimidines, those nitrogenous bases that just simply consist of a single ring of carbon and nitrogen atoms. The purines, of which there are 2, adenine and guanine, abbreviated to A and G. These are bases that have a 2-ring structure of carbon and nitrogen. So we saw a purine, here it is, adenine, in this example. So a 2-ring nitrogenous base there. All right, so let's sum up what we've learned so far. So in the nucleic acid called DNA, the monosaccharide, or the sugar, is always 2-deoxyribose, which simply means that the OH, that is the hydroxyl, at the number 2 position in that sugar is replaced simply by a hydrogen. Okay, let's look at that. So if it was 2-deoxyribose, here we go. So here's the number 2. This OH, which is called a hydroxyl, that would be simply a hydrogen if the sugar was 2-deoxyribose. All right, the bases and therefore the nucleotides that are found in DNA are adenine, thymine, guanine and cytosine. Now the nucleic acid called RNA has a ribose monosaccharide, not a 2-deoxyribose monosaccharide, and the bases and therefore the nucleotides that are found in RNA are adenine, uracil, so there's no thymine in RNA, there is a guanine, there are guanines and there are cytosines. So getting back to DNA, DNA exists naturally as 2 long chains of nucleotides running in opposite directions to each other. and wound together in a double helix formation, which we'll see here on the next slide. Yes. All right, so this is the structure of DNA. This one is a little bit hard to follow, so let's look at this one here in B, this representation here. So 2 strands of DNA, 1 strand in pink, 1 strand in purple. Okay, they are bound together, they are bonded together, I should say, by special bonds, and then they are twisted into this double helix formation. All right, let's look a little bit more closely. Let's just take this boxed region here, which consists of 4 nucleotides, 2 on one strand of DNA and 2 on the opposite strand of DNA. Enlarge that, and this is what we see. And now we can really see the chemical structure of these 4 nucleotides and the way that they're bonded to each other within the one strand and the way that those 2 strands are actually bonded together. All right, so let's start by just looking at these 2 nucleotides here on the one strand of DNA. So remember, here we're looking at the opposite strand. All right, so let's look at this strand of DNA. And what we're looking at here is 1 nucleotide, which we call cytosine, because it has a cytosine base. And this second nucleotide that it's bonded to is called thymine, because it has a thymine base. Now, these nucleotides are all bonded together in one very, very, very long chain. When the 2 chains bond together to form one molecule, that's then referred to, or eventually, after further sort of coiling and twisting, it's referred to as a chromosome. All right, so what are the bonds that actually hold or bind nucleotides together within the one chain? Those bonds are called phosphodiester linkages. Don't worry about what that means for now. But all you need to know is that these are the bonds, phosphodiester linkages, that bind nucleotides together in the one chain. All right, now we can see also that there are distinct ends to each chain. So this end here is referred to as the 5' end, and this is where the phosphate is. And down at this end of the chain, this is referred to as the 3' end. And you can see that that 3' end comes from the actual sugar of this thymine nucleotide, in contrast to the 5' end of the chain, which refers to the phosphate of this cytosine. All right, let me clear that and let's focus now on the opposite chain. All right, now you can see something really odd here in that the nucleotides appear to be upside down relative to the other chain. So here we have the guanine, a guanine nucleotide, and now we have here the 3' end, okay, rather than the 5' end. And it's the 3' end because we're referring to the sugar of this guanine nucleotide. That guanine is bonded, so there should be a bond in here. That guanine nucleotide is bonded to this adenine. And once again, we have the 5' end down here, which is opposite to the 3' end here. And of course, that's because the 5' is referring to that phosphate group. All right, once again, those nucleotides are joined together by what's called a phosphodiester linkage. But now let's look at what binds those 2 chains of nucleotides together, those 2 strands of nucleotides together. Here, in between the nitrogenous base of the guanine and the nitrogenous base of the cytosine, we have a bond that's formed right there. Those dotted lines, that dotted line there and there And here we have 3 bonds that always form between a cytosine on one strand and a guanine on the other strand. Always those 3 bonds, and they are called hydrogen bonds. We'll talk about them in a later lecture. Between adenines and thymines, we have the same bonds, but only 2 of them. Only 2 hydrogen bonds that hold those DNA strands together. And that's why when there's a cytosine on one strand, it will always bond to a guanine in the opposite strand. When there's a thymine on one strand of DNA, it will only bond to an adenine in the opposite strand. And that's because of the number of hydrogen bonds that can be formed between those neighbouring nitrogenous bases. So they're hydrogen bonds and phosphodiester linkages in DNA. So let me clear that. And we have completed tonight's lecture. So in summary, organic molecules are composed principally of atoms of 6 elements: carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur. By the way, I'll get to that. Biomolecules include 4 types of small molecules. We have the sugars or the monosaccharides, we have fatty acids, we have amino acids, and we have nucleotides. Really important unit of molecules that we looked at there. Oh, a bit of feedback there. All right, the most abundant biomolecules in living organisms are the carbohydrates. But if we just think about any type of molecule, what would be by far the most abundant molecule? So we're no longer talking about just biomolecules, just any type of molecule. What would be the most abundant molecule in any living organism? Any idea? Carbs, yes, the most abundant biomolecules, but what about just considering any type of molecule? It doesn't have to be a biological molecule. Water, someone's got it? Water is by far the most abundant molecule in living organisms, correct? Okay, so we've looked at lipids and the fact that they are predominantly hydrophobic, but of course we have those amphipathic molecules such as phospholipids that are exceptions to the rule. We've looked at proteins, which start off as polypeptide chains of amino acids. They are linked together by peptide bonds. They will form secondary structural motifs such as alpha helices and beta sheets. Then they will form the tertiary structure and finally the quaternary structure to form the final functional protein. We also looked at nucleic acids, specifically DNA, which is the molecule in which information is stored and passed from generation to generation. But we also touched on the other major type of nucleic acid, which is RNA. And we won't talk about what RNA does now, but we certainly will in a later lecture. So well done, anyone that's gotten through all that, which is most students. I think we only lost 2 students from the class. So well done. That is a big night. That's as difficult as it gets, in my opinion. So it will take some revision on your part to really get your head around all that, especially if it's the first time that you've actually studied any biochemistry. All right, that brings us to our tutorial questions for this week. These are assessed. They are due next Monday. They are worth 4% in total. Great way to test your knowledge and to make sure that you revise the material and really understand it. Okay, so we've got some balancing of chemical equations that you can practice. We're looking at that term, remember, formula weight and molar mass. So revise those slides before you answer. Then we have our molar mass formula, our molarity formula, molar mass, some rearranging of equations and using two equations to solve one problem. In other words, step one, using one equation, step two, using another equation. All right, then we get into redox reactions. Okay, here are the tricky ones, questions 7 and 8. Okay, so with these ones, don't forget that you need to balance the equations in your answer. And then we have some biological questions on biological molecules. All right, so as I always say, let me know if you get stuck on anything. I can certainly help you most of the time just through email. But if you prefer to chat on the phone, that's not a problem whatsoever. All right, so that's your tutorial questions due next Monday, not next Friday. And now let's look at the prac manual, because obviously all pracs have been cancelled. So the obvious question here is, what are we going to do about prac reports? Now, some students have already done a prac that is very good to write up as a prac report. And let's look at those pracs. Oh, by the way, this prac book is now available on Moodle. Check out the additional information folder and you can find a copy of this prac manual. Okay, so last week, some students managed to do this prac. This is a very good one to write up as one of your prac reports. So those students can certainly write that up as one of the two prac reports. Now, the prac that has to be written up by everyone is this one here, acid-base titration. All right, so given that, the plan that we have is to record a demonstration of both of these pracs. So as some of you know, I've been testing out that GoPro all week, and now we're ready to go. We're going to actually take video recordings of someone demonstrating that prac, carrying out those pracs for you. And that coupled with the results from those pracs will give you more than enough information to write up your two prac reports for this subject. Okay, now the problem with that, so that's very helpful in terms of being able to complete your assessments and complete the subject, but it certainly doesn't help to teach you very important practical skills going forward. So the plan is to run catch-up pracs, not just for living systems, but for a couple of your other subjects. And those catch-up pracs, I can't tell you when they will be scheduled because we have no idea when we can resume face-to-face classes. But at some stage, maybe at the end of the year, or maybe we can incorporate them into second semester. If we can resume face-to-face in second semester, but we want to avoid doing too much of that during second semester because it will be too much of a load, an additional load on students in second semester. So we haven't quite decided yet, and I don't think we can decide for quite a while when to actually schedule those catch-up classes. So obviously, as an example, in Living Systems 1, you learn quite a bit about microscopy. It's not something that you can learn in any way really by looking at a video. It takes practice and lots of practice to become an experienced microscopist. Very important for everyone. All right, so that's something that we would have to consider in a catch-up prac. All right, so questions? All yours.
Hello.
Oh, hello, yes.
Am I able to ask a question now, or shall I wait till the recording ceases?
Oh, probably wait, because we'll take questions about how subjects will be delivered, how practicals will run, any other questions regarding your subjects. Okay, final exams. While you're thinking of your questions or writing them up, final exams, there's a lot of discussion at the moment, a lot of discussion around online, having exams online. So I still can't give you a definite on that. I'm still not sure. As of last week, we could still deliver face-to-face exams, of course, while observing social distancing. But if it comes to the point, which I'm sure it will, where we can no longer do that, where it's no longer safe to do that, then we'll have to consider using what they call proctored exams. Proctored exams, these are supervised online exams where the supervisors basically watch you throughout the whole exam via the webcam. And there's all sorts of rules around what you can have on your desk, what you can have open on your computer, and so on. I mean, that raises problems. You know, what about students that don't have webcams? What about students that don't have the internet bandwidth, you know, to stay basically up and running during a 2-hour exam? But that's what's currently being discussed, not just by Melbourne Polytechnic, but also by La Trobe University. Yes, so any questions about any of your subjects? Hello, Rob. Have you got any questions?
I do.
You do?
I do. Relating to winemaking.
Oh, yes. Yes, sure. Yeah.
Mark's put up a forum topic
right.
on demijohns.
Yes.
And inviting replies, but I'm unable to figure out how to reply in the forum because all it's got in the box there is permalink. There's no reply or anything else.
Oh, that's very odd because the forums, that's the aim of a forum is to allow students to actually communicate with each other and basically have a discussion.
Yeah.
That's a funny one, isn't it?
It is. So is that something in my settings or the way he set the question?
I don't know. I'd have to have a look at that, Rob. But you're probably best to just send Mark an email.
Yeah.
And ask him, just let him know that you can't find any way to respond to that forum.
Yeah.
Yeah. Yeah, and I can certainly have a look too. I can get into that Moodle site and check that out as well.
Doctor?
Hello, Simon. How are you?
I'm well, thank you.
Good, thanks. I will get to your question. I'll just see if anyone else has a question.
Okay.
Yeah, about their subjects. Doesn't look like anyone does, but I'll just give it a couple of minutes. Oh, actually, I might just stop the recording I'm going to stop the recording right now. Stop recording.